yeah. Yeah. Such a good house party, mate. OMG. Yeah. Oh. Nobody here. Benito! Oh, shit! Oh, I think I enjoyed myself too much there. Oh, hello! Yeah, scrap all that DNA technology nonsense, because in this series of videos we're going to be talking about, well, probably the most key, well definitely the most key concept that every biologist should know. So, up yours, molecular. Yes, a subject where biologists don't have to be, looking in petri dishes to earn a living. They can actually go out and enjoy nature. Well, a lot of them do spend a lot of time looking at petri dishes as well, but that just shows how broad this subject is. And hopefully in this series I'll be able to cover almost everything that I've been doing on my course, everything from the origins of life to human evolution itself. Now, just like how I said at the start of my molecular evolution videos, I'm nowhere near an expert on this. This is just my method of revising, and hopefully it's helping you too. I know the molecular ones did help a little bit. Another thing I'd like to say is that I'm extremely sorry for the last episode of Molecular. There was a section where it was just me talking on top of myself and it was an absolute mess. So I've sort of made it a little bit more understandable now. But sorry about that, I blame the editing team. Nothing to do with me, it was the editing team, okay? Okay, I'm going to kick off this series by talking about the basic mechanisms for evolutionary change. And that'll take us through the first few videos. Obviously, we can't go anywhere into that without understanding what the four postulates for evolutionary change are. A postulate is a given truth to a certain concept. So, in our context, it is the four things that must be the case for evolution to occur. One is variability. And what do we mean by that? What we mean is a huge collection of different alleles within a certain population which can be shuffled about and form a whole load of combinations. So you have a continuous variation of certain traits. New traits can be generated through mutations. Now we'll go into all this in a whole lot more detail later on. Two is heritability. Yes, that basically says that genes must be passed on to offspring within the population, so those genes will be retained within that given population. And these genes are inherited independently of other genes. Three is overproduction of offspring. So that's pretty simple. That basically means that the parents produce more offspring than can actually survive. So that is all of aid of increasing chances of survival in the next generation. So you still have a stable population of that species. Four is natural selection, arguably the most important. And that's basically saying that survival of those offspring isn't a random thing. Only individuals which are suited to the particular environment will survive and therefore reproduce and pass on those genes to future generations. So that is to say that individuals with higher reproductive success are better suited to their environment than others. Now, there are two terms I'd like to define before we go any further into this, and that is fitness and adaptations. What is fitness? Well, fitness can mean a whole load of different things in everyday life, but in terms of evolutionary biology, it is the ability to survive and reproduce, hence survival of the fittest. And adaptations are traits that individuals have which increase their fitness, so increase their ability to survive and reproduce. And this is relative to individuals which lack those particular traits. Now here we're going to be talking about postulate 1 and postulate 4 in more detail. Postulates 2 and 3 kind of speak for themselves a little bit, but postulate 1 and 4 require a bit more explanation, so we'll go into the mechanisms and how we get variability within populations and the mechanisms of natural selection. Okay, for the moment then, let's look at the first postulate, variation. What produces variation, what factors promote variation, that kind of thing. Now, in some cases you can tell the genotype, the genetic makeup of an organism, just by looking at it. For example, you can easily tell whether someone has the gene for cystic fibrosis, for example. This is what we call a qualitative trait, where the observable characteristics of an organism, their phenotype, I should be saying, falls into discrete categories. So you either have cystic fibrosis or you don't, okay? And it's these traits which are usually controlled by one single gene. Now, on the other end, we have quantitative traits. One where there isn't discrete categories and you'll get a continuous variation of traits. An example being height. Because for height there aren't any categories which you could fall under. There isn't a small category and a tall category. There's a continuous variation of heights of individuals that are in between tall and small. And it's these which are usually controlled by more than one gene. And it's the interactions between these genes which produce this variation. Now obviously, not all of the observable characteristics that make you aren't completely down to your genotype, your genetic makeup. The environment also plays a massive part as well. And a good experiment to show this was done by Klorenkek and Hisi in yarrow plants. What they did, they took 3 cuttings from the same individual yarrow plant and grew them at different altitudes. And what they found was that plants that grew at lower altitudes actually grew taller than ones that were grown at higher altitudes. So the environment has a massive role to play in determining the extent at which the organism's genome is expressed. But overall, it's thought that about 60 to 80% of the factors that control height are to do with genetics. So the other 20 to 40% is the environment. So your genetic makeup does play a key part in to how tall you are. But what causes this variation? Well, you've probably heard of crossing over before between homologous chromosomes. Well, that plays a massive part in meiosis, when homologous chromosomes get together down the centromere and exchange genetic information from one chromosome to the other. So this builds up different combinations of genes, which leads to greater variation. Remember, quantitative traits. Also, migration plays a role. If you have an isolated population, then another group from another population joins that population, then it will be introducing brand new genes and brand new combinations of genes with them. So the greater the population you have, the greater variation you have, because there is a greater gene pool. However, these two are all to do with just shuffling of genes that already exist. How do you create something absolutely brand new, a brand new trait? And the only true way that brand new traits can be developed is through mutations. So mutations are random changes in the genetic makeup of an organism. But we're not talking about random changes anywhere, they've got to occur in a specific place. And by that, we mean the germline, your bollocks, okay? And it's these gametes which contain the genetic material which can be passed on to future generations, into their offspring. So the mutation has got to arise in the germline for those gametes to contain that mutation, and therefore the offspring to contain the mutation. Now that's generally the case, but in plants it's slightly different because the reproductive structures in plants actually come from somatic cells. Remember, we talk about it a little bit in the molecular series. So any mutations that happen in the soma can be passed on to gametes when those reproductive structures are produced. So how often do these random changes, the mutations, occur? Well, the mutation rate, I've just said mutation rate, what the hell is that all about? Whew, that's better. So the average mutation rate I put up here is on average 10 to the minus 5. That means 1 in 100,000 gametes has a mutation on a particular gene, which doesn't sound very much at all, but when you think of the amount of genes that are contained within a gamete, humans have 24,000 genes in their genome. So you can do a bit of maths here, and you've also got to remember that we're diploids, so we have 2 copies of each chromosome, and that mutation can occur on either one. 10 to the minus 5 is the mutation rate. If you times that by 24,000, that means you'll get 0.24 mutations per generation, but then times that by 2, because we've got diploids, so that's 0.48. So that means, if you do a bit of maths, that in a population of 100 people, you'll get 48 new mutations arising. Which you could think is pretty incredible, really, and it is. But what effect do these mutations actually have? Well, some can have some really drastic effects. There is a trait called antennapedia in Drosophila flies, which means that the antennae of the fly are replaced by its legs. So you get fruit flies with legs sticking out of their heads. That's pretty drastic. Must be an absolute nightmare for them to get their pyjamas on at night, I've got to say. The fact is that most mutations that are produced are neutral. They don't do anything. It's either here or there. You also get some deleterious ones. A very small proportion of these will actually be beneficial to the organism. Likewise, very few will actually be lethal. I think if you say the word mutation to a, average person, they'll be like, oh no, that's bad. Well, they're not really. That's just because you've been watching too many sci-fi films. In particular, where mutations are likely to have an effect is on pleiotropic genes. What's pleiotropic genes? A pleiotropic gene is a gene that affects more than one trait. So if you have a mutation in one, it's likely to have an effect on more than one characteristic of that organism. An example of a pleiotropic gene is what's called the PER gene in Drosophila flies, and this gene controls circadian rhythms, and it also controls wing movements during courtship. So quite a crucial gene, really, for those lovely little fruit flies. So let's talk about the types of mutations. Well, you can have point mutations. That's mutations that occur at a single base. Now remember, basic biology here. On DNA, you've got your 4 bases. You've got A, G, T, and C. A base pairs with T, G base pairs with C, and vice versa. I've arranged this in a particular order because the A and G are your purine bases, and the T and C are your pyrimidine bases. Now, if an A swapped for a G, or a G swapped for an A, likewise, if a T swapped to a C, or a C swapped to a T, then we call this a transition. Okay? It's just going from purine to another purine. Okay? If, however, we went from a T to an A, or a C to a G, or a G to a T, or a C to an A, the amount of combinations is, a lot in this case. That's called a transversion. Right, isn't that the messiest diagram you've seen in your life? Thank you very much. Now, transition mutations are on the whole more common than transversion mutations. After all, your DNA polymerase or whatever it is would have to be pretty stupid to mistake a purine for a pyrimidine or vice versa, wouldn't they? Good God, stupid polymerases. The effect of a mutation, it also depends on where in that gene sequence the mutation has occurred. So remember, triplet code of DNA, and the degeneracy of the genetic code, meaning that one amino acid can have more than one triplet code. The general rule is, if the mutation occurred on the 3rd base in the triplet code, it's unlikely to have much of an effect. And the 1st and 2nd is likely to have more of an effect. So the position of where the mutation and the 1st and 2nd is likely to have more of an effect. So the position of this point mutation is important in controlling what amino acid is produced by the end of it. Now we can describe whether this point mutation has had an effect on the protein produced by describing them as either silent or synonymous mutations, or non-synonymous mutations. So synonymous mutations are mutations which don't have an effect. That's why they're also known as silent mutations, because nothing happens. So here we have the triplet code for the amino acid glycine, GGC here. And a mutation has occurred to change the C into an A. Now that triplet code still codes for glycine. And that's also an example how a mutation in the third base doesn't really have that much of an effect. However, in this example, we've got glycine, GGC, and the second base has been changed this time from a G into a C, so you get GCC, and that produces a completely different amino acid. So that's a non-synonymous mutation. And this could completely affect the overall product of your gene. The protein structure of the gene produced could be completely different just because of one amino acid. You never know. And what you can do is to measure the ratio of non-synonymous mutations to synonymous mutations to get an idea on the extent of adaptive selection occurring at a particular population. So if it's greater than 1, i.e. there's more non-synonymous mutations than synonymous ones, then we have positive selection. If it's less than 1, it's what we call stabilizing selection or purifying selection. And if it's 1, if the amount of non-synonymous mutations to silent mutations are equal, then we can say that it's neutral, there's no selection happening at all. And I'm aware that I haven't talked about selection at all yet, so hopefully this will make a bit more sense once we get onto that. Now a great example for me to show on what I've just been talking about is in the FOXP2 gene. FOXP2, nothing to do with foxes. What it is, it's one of the main genes which scientists think is what caused the development of language in humans. Obviously language, pretty unique to us humans compared to other animals as far as we know. And we think that it's a mutation in this gene which led us to develop sophisticated language. So here we have a tree diagram of a lot of different taxa, mostly primates, but we've got a mouse here, and for the mouse you have a ratio of one non-synonymous mutation to 131 synonymous mutations. Then you go into the primates, the ratio becomes a bit smaller. You've got a rhesus macaque here, 0 non-synonymous mutations to 5 synonymous. Then for orangutan it's a half, gorilla 0 to 2. But then you have the humans. Now all the ratios before the humans, the ratio was less than 1. So that means all these mutations were being purified out of the population through stabilizing selection that we've just been talking about. However, for humans, we've got 2 non-synonymous mutations for this gene compared to 0 synonymous mutations. So that suggests that there's a positive selection for these mutations that have occurred in the FOXP2 gene. And it's been seen that individuals which have other mutations, where these mutations occurred on the FOXP2 gene, actually have some quite serious speaking defects. So it is very likely that this gene plays a key part in us being able to speak. Now there are other factors which increase the amount of variation within the population. Oh, that rhymed. First one is heterozygote advantage. So that means it's advantageous to be a heterozygote. So on each homologous chromosome pair, you have a different allele. Now the absolute standard textbook example for this is the relationship between sickle cell anemia and malaria. If you haven't heard this and you do biology, you must have been living under a rock, seriously. Okay, there are 2 hemoglobin alleles that you can possibly have. One is the good one, so the one for normal hemoglobin. We'll give that the letter A. And then there's the mutated allele, which is what causes sickle cell anemia, causes the red blood cells to go that weird sickled shape and not work so well. I'm going to give that allele an S. So obviously there are 3 possible combinations. You can be homozygote, you can be heterozygote, or you could be homozygote for the sickle cell allele. Surely you might be thinking that it's best to have the homozygote normal hemoglobin allele. And that's kind of true, I guess, because you haven't got any sickle cell genes in there at all. Unless there's malaria going round. Now the malaria parasite, the Plasmodium protist, loves to infect nice, pristine, nothing wrong with them, red blood cells. Now you can see where we're going here. So if you are homozygote and you get malaria, you're in trouble, basically. However, if you're a heterozygote, you still have normal blood cells, just like a homozygote, but if there's reduced amounts of oxygen within the red blood cell, the cell will begin to sickle, be sickle-shaped. And so that means when the Plasmodium infects the red blood cell, it'll take away the oxygen, causing that red blood cell to die. And whilst doing that, it will kill off the malaria parasite. So it's like killing two birds with one stone, basically. If you're homozygote A, then you haven't got sickle cell anemia, but you're likely to get malaria. And if you're homozygote S, then you've got sickle cell anemia. So it's curtains for you, I'm afraid. However, if you're heterozygote, then if the malaria infects you, the cell will sickle, killing the parasite, and it's all happy days. You can carry on living. Another promoter for variation is something called frequency-dependent selection, and the best way to explain this is an example using scale-eating fish. Right, I'm going to do some great impressions of fish now, you're going to love them. Now there's two types of scale-eating fish. Ones which point their mouths to the left, like this, and ones which point their mouths, you guessed it, to the right, like that. And this is my host fish, so the fish with the scales that me, the scale-eating fish, wants to eat. Okay? So, if I'm a left-facing fish, like that, I can only feed off the scales from the right side of the fish, can't I? Okay? Now, if I'm a right-mouthed scale-eating fish, like that, I can only feed off the left side of the fish. Looking at it this way. I'm not getting my left and right mixed up, I promise you, okay? Now this host fish is more likely to defend the side with the most scale-eating fish on it. Because think about it, if you've got 10 people pestering you on your right and 1 on your left, then you're more likely to shove off the people on your right first, right? Okay, so here's a load of scale-eating fish feeding on the scales on the right side of the host fish. One's a little beak like that. I do apologise for the drawings, by the way, they look a bit like sperm, but never mind, we can live on. Now, this guy here has the whole of this side of the fish to himself, and what's more, the host fish is more likely to defend the side with the most scale-eating fish on it. So this guy's got it sorted, basically. He's living the high life right now. So he is the one that's more likely to survive and reproduce. So therefore, this will create a load more fish with their mouths pointing to the left. So then, you'll get more fish going to the left side. And the cycle starts again. This guy here on the left side of the fish has an advantage, so that means that allele is favoured compared to that one. And what you'll get is the frequency, the allelic frequency, oscillating between the two different mouse morphs constantly. We call this oscillating selection. So that means both alleles are conserved, adding to the variation. There are a number of different factors. There's hybridization that can lead to new phenotypes. This is particularly noticeable in plants which undergo a lot of hybridization. Something called polyploidy. We'll talk more about polyploidy in a later video. There's also horizontal gene transfer. This is where species share pieces of DNA. So DNA from one species can go into the genome of another. And this can be done during virogenes, so sort of RNA viruses which can go into DNA using reverse transcriptase and incorporate themselves into the genome. Okay, enough of that now then. Now, I've mentioned the word selection quite a bit already, so it seems only right to move on to explain what natural selection actually is. Now there is a plethora of different ways in which you could describe natural selection. One of them is differential reproductive success. So, like how I said earlier, it's the fact that survival and the ability to reproduce to give successful offspring isn't random. It's the individuals which are better suited to their environment that are able to survive. And this can't be done without variation, because we need a large gene pool for different organisms to have different characteristics. If there was no variation, they'd all be the same, so there would be no way natural selection could happen. But of course, one of the results of selection is that it changes the allele frequency within the population. So let's go straight in to talk about the different types of selection. Now, in quantitative traits, the distribution of phenotypes is likely to be normal. It follows this normal distribution curve that we can see here, this bell-shaped curve, where the mean trait is likely to be at the top of the curve here. So here, this is the phenotype, the mean phenotype of whatever trait we're talking about. So on the y-axis here, we've got the probability of that particular phenotype occurring. So that graph makes sense. Now, there are three types of selection in nature. One is slightly less common than the others, but we'll go through it anyway. There is directional selection, and that favours extreme phenotypes. And that's represented by these two graphs here, which basically say the same thing. Now, let me think of an example. An example of an extreme phenotype would be extra long, extra thick fur if temperatures suddenly dropped in a mammal, let's say, I don't know, a mouse. So, in that selective pressure, these extreme cold temperatures, that will favour mice which have extremely long fur, the absolute extreme of that phenotype range. Now let's say this normal distribution curve is now fur length, and at colder temperatures selection will favour bigger fur length. So what will basically happen is that this normal distribution curve will shift to the right, okay, because on the x-axis we've got increasing fur length now, obviously. So what will happen, the shape of the normal distribution curve won't change, the variation will stay the same, it will just get shifted. So the mean phenotype now is in a different place to where it was before. And what we can see here in these diagrams is that phenotype increases, probability of survival increases. Likewise, if let's say if it was really hot, then it will favour mice with smaller fur lengths, let's say. So here 0 is the extreme, and the probability of survival with increasing fur length here slowly decreases. Okay, that's the first one. The second type is stabilising selection. Now, stabilising selection favours the mean phenotype, as you can see here. Now, I've tried to show in this diagram here is that this normal distribution curve is starting to be a bit thinner, because any traits which are distant from the mean phenotype, those individuals have a disadvantage. So let's say it's perfect temperature out in the British field. There's perfect temperature, so that means mice with longer fur are probably going to get a little bit hot under the collar and probably die of hypothermia, therefore not surviving to reproduce. How jolly. And ones with really short fur likely get a bit cold, so might get a bit of hypothermia and freeze their bollocks off, okay? So the frequency of these phenotypes decreases, so the probability of survival drastically increases when it's around the mean fur length here. So we've basically got a normal distribution curve again. Now the third and final form is a little bit less common. This is disruptive selection, and this is where it favours extremes on both ends of the spectrum. Now an example of this is, I'll read it to you, is the black-bellied sea cracker. Now this is a type of finch, I believe, and there are two morphs. Ones with really small beaks and ones with really large beaks. Now these two morphs, they're the same species obviously, but ones with small beaks feed on smaller seeds and ones with bigger beaks feed on bigger seeds. And having these two morphs is quite useful because it prevents intraspecific competition. between the two species. So there's less competition for food within the population of these finches, because half of them will have short bills and the other half of them will have big bills. If you've got something in between, then your bill isn't really well suited for any kind of seed. And maybe, this is just me hypothesising here, it may reduce your chance of finding a mate because that's less attractive to females or something. So I don't know. So this is the relationship you see. We have phenotype going on the x-axis, probability of survival, greater survival when low beak depth, and greater survival when there's high beak depth. In between, not so much. So if you have in the population loads of mutations happening, creating new alleles, new traits, new phenotypes, and you've got selection occurring, then that really is a potent evolutionary force, a sign that your population is evolving. Now, of course, Darwin, when he published On the Origin of Species in 1859, he didn't know anything about genetics. Really, we didn't know about DNA, we didn't know how things were inherited, and so on. It was actually Mendel, who was doing his work on peas at more or less the same time as Darwin, that provided the basis of modern genetics. So if we link Mendel's work with Darwin's work, we can create something called the modern synthesis, which shows that evolution over time is caused by small genetic changes between individuals. Now these gradual genetic changes between individuals in a species is what we call microevolution, and as that carries on, we can see how that can lead to macroevolution, the evolution of more taxa, new taxa, so speciation, basically. Now we're going to be talking more about speciation in a later video, so we'll leave that at that for now. One real life example that's actually going on at the moment is caused by deforestation in Brazil. And this is a really good example on how us humans are affecting selection pressures and success, reproductive success within other species. Basically, a lot of deforestation is going on in Brazil, as you might imagine, for a whole load of different things. So loads of rainforest is being destroyed. Buggers. But anyway, let's not start crying just yet. We're talking about toucans here, that's very nice. What's happening is that large frugivorous birds with really large beaks are in decline. No, the toucan's in decline now. Everything's going wrong. Now it's the toucans and toucanettes with their large stout bills that are able to crack open the very largest of seeds. Now, with less toucans in the area, that means less large seeds were going to be dispersed. So that means the proportion of plants which produce large seeds in the next generation will be less. So that means selection will favour plants which produce smaller seeds, because smaller seed dispersers weren't affected by the deforestation. So we're talking about things like finches here. Their populations stay relatively constant. So these are still dispersing smaller seeds with no problem at all. Now, for the plants, having a smaller seed brings a disadvantage. With a smaller seed, then you've got less nutrient reserves within that seed, which allows the seed to survive and as a result germinate. So germination success is reduced because selection has favoured those with smaller seeds. And these results have been shown in a study which was conducted over 75 years, which, when you think about it, is quite a short amount of time. And that's it, there's your introduction to this series. Hope you enjoyed this, I hope you watch more that will come up. We're going to be talking about species and taxonomy and all that next time, so I'll see you then.